Hello, I'm Clarisa Castaños. I'm doing my PhD with the CRC for honeybee products. I will talk to you about my research in honeybee nutrition, that is specifically about the early detection of malnutrition and colony collapse. First, I will tell to you a little about me. I come from Mexico. I studied a bachelor's degree in biology in the National Autonomous University of Mexico. Here, I did my thesis in molecular science about genetics and DNA regulation of bacteria. But then I work with insects. I do some kind of work physiology and ecology with damselflies and also water bugs. Then I want to continue working with insects, and I came to Australia to do a PhD in the University of Western Australia. Here, I joined the CRC for honeybee products in the honeybee health research group of Dr. Julia Grasso. And here I'm working with honeybee nutrition using molecular science. I will talk to you about this. Okay, a little about honeybee nutrition. A good nutrition is indispensable for the optimal performance and productivity of honeybee colonies, as well as for avoiding and resisting disease and parasitic infections. The nutritional sources of honeybees are nectar and pollen, and they transform these into honey and bee bread. The nectar is the primary provider of carbohydrates for bees, and carbohydrates are the immediately source of energy for the metabolism. They are important for processes like regulating the body and hive temperature or for provide food for flying. When there is a decrease of the nectar consumption, the colony becomes more aggressive and reduces the foraging and hygienic behavior. On the other hand, from the pollen, bees obtain mainly proteins, amino acids, and lipids. For example, proteins are used for brood rearing, so they are essential for the proper development of the brood and for promoting the laying of eggs from the queen. Lipids are the long-term storage energy reserves, but they are also a signal of working shift behavior in the colonies. So, a lack of pollen causes decrease in the amount of brood and a rapid decline of the colony size. In the Australian beekeeping industry, bees provide pollination services of crops, and they are especially important for the production of valuable monofloral honey. But to maintain the healthy nutrition of the bees and achieve an optimal productivity of these services, beekeepers need to move their hives hundreds of kilometers, chasing for different floral sources. Or if it's necessary, they supplementally feed their hives, for example, in case of bushfires or when there are delays in the flower season. Therefore, it is essential for the beekeepers to be able to tell the optimal feeding timeframes for their hives, as well as the quantity and the quality of food that they need to provide to achieve the best productivity. All of this depending on the energy and the time, and also on the money they want to spend on it, or they can do it. Related to this comes the aim of my project, that is basically to detect when bees are in a nutritional decline. This by evaluating the molecular profile of the decline and correlated with hive indicators that beekeepers can detect in their hives. In that way, I am defining metabolomic and lipidomic profiles of bees during and after nutritional stress. I am determining molecular markers for the early onset of starvation. Then, with these techniques and the molecular markers I want to define, I will evaluate different kinds of supplementary food. This is specifically for the building or recovery of struggling hives. All of this will help to define management strategies to prevent and control bee malnutrition, achieve an optimal productivity, and provide food security. To explain you a little more about how I'm doing this, to define these molecular profiles, I have done a field experiment where I provide 3 different diets to 18 hives. I had a starving group that I didn't provide food, a sugar group, and a sugar and pollen group. That is the best nutrition for honeybees. We controlled the food we provide to these hives during 4 weeks, and then we let the hives to recover with free foraging. This is me setting up the colonies. We set up clean new boxes with new friends, and we put bees from different hives. After building the colonies, we shift them to a redcom site to allow them get strong in a good nutrition before starting the experiment. After this, we moved them to this unproductive site without food sources, so we could control the nutrition. We did the sample of the hives each week during 4 weeks, so we went once a week. And first, we take measures of the hives. Here we take the measure of temperature, humidity, and weigh the hives. Then we take photos of every frame in each hive to measure the quantity of brood. We take samples of the bees to do the biochemical analysis later, and at the end, we feed the colonies placing sugar syrup and pollen patties in the corresponding hives. After this, for keep tracking the recovery of maintenance of the nutrition of the hives, we kept shifting the hives looking for good honey flows, stopping the differential feeding and allowing the free foraging. Okay, as high performance results, here I have some of the measurements we take. First, I have the weight, the quantity of brood, and the temperature. These are the weeks we were measuring. Before the 4 weeks we give the diet treatments, and after this, we went to the honey flow. For example, for weight, we can see here that the red group is the starving group. And it's obvious how between the 0 and the 4 week, the weight decrease in the starving group. But after we shift the bees and we get them to the recovery side, they never recovered the condition again. So after the 25 week, they were still struggling. For the brood, it's a similar story, but then they recover at the 6th week. And here in temperature, we can see how it's very constant until the starving group is really struggling and doesn't have food. Then the temperature is dysregulated. They cannot control the temperature in the hive. Then to correlate this with molecular analysis, for the biochemical analysis, I use the fat body organ of the bees. I dissect all the bees collected from the 18 hives during the 9 weeks, taking 20 bees per sample. I crushed the bees and I did extraction of the different macromolecules I wanted to analyze, that were metabolites, lipids, and fatty acids. Then I processed my sample through this machine called Mass Spectrometry. After this, each compound that the machine detects can be visualized as peaks, so I can see the presence or absence of difference in abundance of the compounds found on my samples. Here we can see how the starving group has very low abundance for this compound, then it increases in the sugar group, and it's very abundant in the sugar and pollen group. This represents just a compound. Here are some results of my lipidomic analysis, and after processing the data with different softwares, some compounds that change in abundance between my treatment groups and weeks could be identified. For example, in this case, I identified 2,600 relevant compounds that are changing between my groups, and 1,600 were identified by name. In this plot, you can see how, when considering the change in abundance of these compounds, my groups are clustering together and apart from each other. This clustering is good because it's telling us that each compound is behaving different depending on the treatment. This is a heat map that shows the difference in abundance of subcompounds between just the starving and the sugar and pollen treatments. The color in the left are different kinds of lipids, and in the bottom we have the difference in the weeks, from the week 0 to the week 8, and it's clustering together the weeks that are similar. Here we can see that the week 3 and 4, when the starving group was really starving in a bad nutrition, are clustering together. And about the compounds that were detected, it's more red when it's downregulated and dark when it's upregulated. So we can see that for the time 3, many compounds are being downregulated. This is good because it's before the really starving moment. So the bees were really struggling here and the quantity of lipids was decreasing. These are some examples of the lipids that looks like a good biomarkers. Here we have some plots of them. Again, in the bottom we have the weeks, and in the left is the abundance of these lipids. This is the difference between the sugar and pollen group and the starving group. We can see how in most of the cases in these three plots, the lipids are decreasing, and it's a good change sometimes in the time 3. This is before the time 4, that is the really struggling moment when the starving group was in a very bad nutrition. This represents that these lipids could be good biomarkers to tell us the early onset of the malnutritional state. Okay, as a resume of what I have done and what I'm doing, I'm evaluating the molecular profile of bees under nutritional decline, and I'm defining these biomarkers for the early onset of the decline. Then I am correlating these with high performance indicator. What is next is to evaluate supplementary food to test the best feeding timeframes and food sources in terms of artificial feeding or pollen sources of feeding, both or supplementary feeding. We want to test these different supplementary food for the building of colonies previous a productivity event like honey harvesting or pollination. All of these to add to the principal outcome that, just to remember, is to define nutritional strategies to prevent and control malnutrition in honeybees, in this case, using biomolecular techniques. I want to acknowledge all the people that is guiding me and is helping me in this project, mainly my supervisors and the beekeepers involved. Also, I want to acknowledge the Honeybee Research Group and the CRC for honeybee products. Well, if you have any questions, feel free to ask. Thank you.